a new DNA test developed by researchers at the Garvan Institute of Medical Research in Sydney and collaborators from Australia, UK and Israel has been shown to identify a range of hard-to-diagnose neurological and neuromuscular genetic diseases quicker and more accurately than existing tests. The researchers say that they correctly diagnosed all patients with conditions that were already known, including Huntington's disease, Fragile X syndrome, hereditary cerebellar ataxias, myotonic dystrophies, myoclonic epilepsies, motor neuron disease, and more. The diseases covered by the test belong to a class of over 50 diseases caused by unusually long repetitive DNA sequences in a person's genes known as short tandem repeat STR expansion disorders. They are often difficult to diagnose due to the complex symptoms that patients present with, the challenging nature of these repetitive sequences, and limitations of existing genetic testing methods. The study published in the journal Science Advances shows that the test is accurate and allows the team to begin validations to make the test available in the pathology services around the world. A patient who participated in the study, John, first realized something wrong when he experienced unusual problems balancing during a ski lesson. John says that he had test after test for over 10 years and absolutely no answers as to what was wrong. He was eventually diagnosed with a rare genetic disease called canvas, which affects the brain. He says that it is exciting to know that in the near future, others with these types of conditions will be able to get a diagnosis quicker than he did. The researcher says that for patients like John, the new test will be a game changer, helping to end what can often be a taxing diagnostic odyssey. Repeat expansion disorders can be passed on through families, can be life-threatening, and generally involve muscle and nerve damage, as well as other complications throughout the body. Quicker, more accurate diagnosis for patients avoids diagnostic odyssey. Current genetic testing for expansion disorders can be hit and miss. When patients present with symptoms, it can be difficult to tell which of these 50 plus genetic expansions they might have. So, their doctor must decide which genes to test for based on the person's symptoms and family history. If that test comes back negative, the patient is left without answers. This testing can go on for years without finding the genes implicated in their disease. The researchers call this the diagnostic odyssey, and it can be quite stressful for patients and their families. This new test will completely revolutionize how we diagnose these diseases, since we can now test for all the disorders at once with a single DNA test and give a clear genetic diagnosis, helping patients avoid years of unnecessary muscle or nerve biopsies for diseases they don't have or risky treatments that suppress their immune system. Although repeat expansion disorders cannot be cured, a quicker diagnosis can help doctors identify and treat disease complications earlier. Using a single DNA sample, usually extracted from blood, the test works by scanning a patient's genome using a technology called nanopore sequencing. By unraveling the two strands of DNA and reading the repeated letter sequences, combinations of A, T, G or C, the researchers can scan for abnormally long repeats within the patient's genes, which are the hallmarks of disease. The nanopore technology used in the test is smaller and cheaper than standard test, which the team hopes will smooth its uptake into pathology labs. With nanopore, the gene sequencing device has been reduced from the size of a fridge to the size of a stapler and cost around $1,000 compared with hundreds of thousands needed for mainstream DNA sequencing technologies. The team expects to see their new technology used in diagnostic practice within the next 2 to 5 years. One of the key steps towards that goal is to gain appropriate clinical accreditation for the method. Once accredited, the test will also transform research into genetic diseases.